student. On the behalf of KTHM College, Nasik and Department of Chemistry of this college, I am Dr. R.K.Pawar welcomes you in this M.Sc.II. Inorganic Chemistry class. Dear student, in this lecture we are going to discuss the Metalloenzyme of copper, Azurin. We know that the Metalloenzymes are nothing but the metal containing enzymes. Enzymes play a very important and vital functions. As like also in previous lecture we discussed the Metalloenzymes of copper, that is type 1st, type 2nd and type 3rd copper proteins. Now in this lecture we are discuss the structure, enzymatic mechanism and function of Azurin. So this is the structure of Azurin. In that you can see that copper is in active site. Here there is a, we are just writing here the binding atoms of the respective ligands here. So this oxygen is from glutamine and these two nitrogens are from the histidine 206 and histidine 196 residues. Here two sulfurs are from the cysteine and this is from the methionine. So here this is the structure of Azurin. This is the oxidized structure of Azurin. In that exact we have to see the whole ligand structure. Instead of this, in this structure we are just writing the first ligand atom here. The introduction of Azurin. In introduction, we know that the Azurin is a small periplasmic copper containing bacterial electron transfer agent. So in introduction we can say that Azurin is a small periplasmic bacterial, because it is found in bacteria generally, or it can transfer electron in bacteria Pseudomonas. Bacterial blue protein, it have a blue intense color. It is found in bacteria Pseudomonas. Actually in bacteria it can transfer single electron. So in bacteria it transfer the single electron in cytochrome chain. In cytochrome chain. So this bacteria is in oxidized form, that is copper 2 plus. We know that in bacteria there is a copper is an active site and this copper is oxidized and reduced by transferring electron. In oxidized state the copper is in +2 and in reduced form it is in +1. So in conversion between the +2 to +1 and again +1 to +2 there is a single electron transfer in between this reaction. So now the fourth point is that cytochrome chain it undergoes oxidation reduction by copper second to copper first here. So this is a general introduction of Azurin. Now let's discuss what is the structure of Azurin. say that in this structure, this is oxidized form of azurin actually, and this is a azurin, and this is a structure. In that we have written just the ligand containing atoms attached directly to the copper. So here there is a glutamine 2 histidine, that is a methionine of sulfur. So this is a methionine of sulfur, and here again there is a cysteine sulfur attached to the copper. Now azurin is a tetrameric protein. So azurin is a tetrameric protein which have molecular weight, each monomer that is 14 kilodalton, and it is composed of 128 amino acids are composed in azurin amino acids. Now if you can observe the structure, here that is a trigonal bipyramidal geometry is observed in azurin, and here there is a, if join it, then you can say that this is a equatorial and this is two are axial ligand. So here there is a distorted geometry of the azurin, that is a distorted trigonal bipyramidal geometry is observed in azurin, where the at equatorial position there is a 2 histidine and 1 sulfur of cysteine. Here at axial position there is a one is a glutamine acid residue, carbonyl group of glutamine acid residue, and second one is a methionine sulfur is attached. So the strands are connected by turns and alpha helical insertion also. So we can say that the strands are connected by turns and single alpha helical insertion. Now the next one is that at azurin, in azurin the 3 ligands, that is 2 histidine and 1 cysteine residue are at equatorial position, whereas the 2 axial ligands the carbonyl oxygen of glutamine and methionine residue are at axial position, which is weakly bound in that position. So the geometry of that azurin is a distorted trigonal bipyramidal. Here we can say that these two axial ligands are not in equidistance to each other, or don't have a proper angle in between them, so the structure of that azurin is a distorted trigonal bipyramidal geometry is observed. So the geometry of azurin is distorted trigonal bipyramidal here. So this is a general structure of that azurin. There is a two hydrophobic patch, and this hydrophobic patch there is a two residues, that is a methionine and the glutamine acid residues are present at an hydrophobic patch, which are interacted with their redox partner, that is a cytochrome and nitrite reductase. So in structure we can also say that the azurin have a large hydrophobic patch. Hydrophobic means repellence of water. Hydrophobic patch. In this patch, these two residues, that is methionine and that we can simply write an abbreviation, that is MET, methionine, and glutamine, that is a GLY, glutamine residue are present, and these residues are interacted with to their redox partner. These two residues are interacted in hydrophobic patch in their redox partner, that is a cytochrome and nitrite reductase. So this is a overall structure of this azurin reactions or enzymatic mechanism of azurin. Reaction of azurin, that is a enzyme mechanism. So we know that azurin in its oxidized form receives an electron from its redox partner and goes to the following reaction, that is this reaction is, so we can say that azurin can accept electron, that is in oxidized form, from its redox partner and reduce as like following reaction. So here if we see that the copper is in plus to oxidation state, Az is azurin, As an abbreviation is used for Azurin in whole reaction, that is short form. So Azurin, and then there is a electron transfer. So here the copper is in +1 state, that is a reduced form, and here that is an oxidized form. So here we can say that copper 2 plus is reduced to copper +1. Here Azurin takes an electron or accept electron from the redox partner and reduce is like the reactions are taking place is as like, and the redox potential for this reaction is a redox potential is approximately 310 MV. So this is a general reaction or enzymatic reaction of Azurin. Now actually the beta sheet of Azurin is strongly coupled with the redox center, electron transfer redox center, and these reactions are generally observed. So let's discuss the biological function of Azurin. As we know that the Azurin is a copper containing blue copper protein. It is a very intense color. It can transfer a single electron in cytochrome chain. In that, when there is a transfer of electron, the copper oxidized to reduced copper first, copper second to copper first, means this is an oxidized to reduced copper. So let's discuss the main biological functions of Azurin. So first, Azurin function of Azurin. So here you can write Azurin transfer single electron in cytochrome chain. Again, the second main function of Azurin in nitrogen fixing organisms. In nitrogen fixing organisms, Azurin serves as electron transfer agent, or electron transfer donor to nitrate reductase. Means that Azurin also play a very important function in the nitrogen cycle. Now the next biological function is that Azurin supports oxidative deamination of proteins. So this is one of the most important function of Azurin, that Azurin support oxydeamination of primary amines by passing electron from aromatic amine dehydrogenase cytochrome oxidase. To cytochrome oxidase. Now from this cytochrome oxidase, it can again transfer to cytochrome C type protein to nitrite reductase. So we can say that for again it can transfer to from cytochrome C to nitrite reductase. So these are the main important basic functions of azurin. That azurin can transfer single electron in cytochrome chain. It supports the deamination of primary amines. So these are the main important biological functions of azurin. So we will stop here. In next lecture we are going to discuss the next metal enzyme of copper, that is a plastocyanin. Thank you.